Welcome back for this last talk on equilibrium, where we're going to look now about static equilibrium and linear acceleration. So it has nothing to do with rotation. It has to do with the head is tilted one direction or the other direction, whichever way it's going to be tilted. And so static equilibrium is often called gravitational equilibrium because the receptors are sensitive to gravity, okay? So when we are talking movement in the vertical plane, as if you were looking down at your shoes or up at the sky, so just think of the two parts for saying yes, not that you're moving, but you're static, okay? Those are going to involve mechanoreceptors located in the saccule, the little part of the membranous labyrinth that's closer to the cochlea. In contrast to that, if you're doing horizontal plane movement, like you were saying, no, okay, that will involve mechanoreceptors in the utricle. So should we get started? Inside that vestibule, the larger utricle was closer to the semicircular canals, the smaller saccule closer to the cochlea. I know I've said it multiple times. I just want to make sure you're well oriented to where we are. Inside both the utricle and the saccule, there is this pale purple lilac area here. And this area is called the macula. And this is where the mechanoreceptor is located. So both the utricle and the saccule contain hair cells. And these hair cells are embedded in that macula. And so they're going to look the same in both. And here's how they look. We have those epithelial cells, then we have the pink hair cells. The hair cells have all these stereocilia. There's even more here than there were earlier when we were talking about hearing. There's at least 70 stereocilia in this place. And the kinocilium is the longest one. And if you look here, you can see that they're all lined up from shortest to longest. Now, what happens here is kind of like a combination of everything we've already learned. There is something called the otolithic membrane, and the membrane does not sit on top. The membrane completely covers the apical surface. And the membrane has two components. The gelatinous part, in which the cells, the kinocilium, are completely embedded. They're in the jello. And then sitting on top of the jello, there's a whole bunch of microscopic calcium carbonate crystals. And these crystals are known either as otoliths, which basically oto is ear, lith is stone, so little ear stones, or they're also known as statoconia because the otolithic membrane was previously known as the statoconic membrane. But I think otolithic is just a lot easier word than statoconia. All right, so that is the setup. The membrane is constantly pushing down on these cells because here we have this cut, but we have pressure coming from the stone sitting on top of it. So when you're upright, that direct pressure, there isn't that much stimulation. Everything is sitting upright. Everything is hunky-dory. So pause for a second and review this anatomy before you continue on. Okay, did you have a chance to review it? Good, because now I want you to think. I do a lot of talking and now I'm going to stop and pause because I want you to come up with the answer. Here's the setup in this lady. All right. What is going to happen when she looks down to see if she dropped something on the floor and she's looking at the floor? What's going to happen in this setup? So definitely pause it. Okay, so did you remember there was weight of the stones? What's the weight of the stones going to do to the membrane? So when you tilt, because I guess she's going that way, when she tilts the weight of the stones, what are the stones going to do? yeah, gravity is going to come into play. That's why it's called gravitational equilibrium in some ways. So gravity is going to be pulling those stones downward, okay? And that's going to be pulling the gelatinous membrane. In this particular picture, it's showing it pulling it towards the kinocilium. Don't be memorizing that if it goes towards the kinocilium, you're looking down. If it goes away, you're looking up. Don't be memorizing that. It's just for illustration purposes. Okay. So when the head has tilted, whether it's tilted like this or tilted like this, the otolithic membrane shifts and that bends the stereocilia, and that bending will change the neurotransmitter release, okay? And so if you look at this bottom picture, you can realize that the bending can be going in this direction, or it can be going in the opposite direction. So when the stereocilia are being bent away from the kinocilium, so if it's like this, if the bending is going in this direction, the hair cells are going to hyperpolarize, so they're not going to be releasing any neurotransmitters. So the nerve signal impulses are very slow, few and far between. On the other hand, when these stereocilia are going to be bending towards the kinocilium, those hair cells are going to depolarize, releasing neurotransmitters, and the impulse frequency goes up significantly. Okay. Now, your brain has to do the interpretation of what's happening with the nerve signals, and as part of that interpretation, it's like, what direction of all those directions did my head just tilt? Okay, but remember, if it conflicts with the eye message, we're going to have a bad experience. So let's do the pathway for all the types of equilibrium. Wherever they are, those stereocilia are going to distort. If we're in the semicircular canal, we're in the macula. If we're in the we're either in the macula or the crista ampullus in the semicircular canal and the utricle and the saccule. I said that wrong the first time, sorry. From there, we are going to have our primary neuron carry those nerve signals through the vestibular branch of the which then merges with the vestibular cochlear nerve, goes through the internal auditory meatus, goes inside the skull, and will go to one of two places. I know we've seen it going towards the medulla oblongata. It may go to the medulla oblongata, where it will integrate information from the vestibular apparatus as well as the eye. Okay, is what needs to happen. So in the medulla oblongata, that integration controls to maintain your balance, that muscle contraction to maintain your balance. When it goes, as this light blue fiber is showing here, when it goes to the cerebellum, because some information goes to the cerebellum as well, that's going to coordinate your skeletal muscle coordination that we talked about when we talked about cerebellar function. That also controls your resting muscle tone. From either of those places, we are then going to have our secondary neurons. Well, actually not from either of those places. From the medulla oblongata, we're going to have our secondary neurons then travel to our sensory relay center in the thalamus. And then from there, we will have our tertiary neurons, which actually go to the cerebral cortex for further processing and then make you totally aware of your body position. And with that, we are done with special senses, which means we are finally finished with everything you need to know about the nervous system. So thank you so much for working hard. I know it's a lot of effort on your part, and we really do appreciate it.